Hello everyone. Um, so my part, I'll be talking about the G proteins and the second messengers, and also I'll talk about GPCRs and the regulatory, the regulators of these second messengers. So it's like just a quick recap. The G proteins. What are the G proteins? The G protein is just it stands for the guanine nucleotide. So this G protein they actually exist in three. They are actually heterotrimeric. Heterotrimeric means they exist in three different units. So we have the alpha unit, we have the beta unit, and we have the gamma unit. So that's how they exist. So for the GPCRs, they are. Sorry, could you go to the next slide, please? Uh, So it begins with a selective detection of surface receptor by extracellular ligand. So what this slide is trying to say is for the second messenger pathway, how it all begins. It begins with the ligands. Once the ligand binds to the receptor at the extracellular part of the cell. Now, this ligand could either be neurotransmitters or hormones. So once this ligand binds to the ligand, once it binds to the receptors, what happens is it brings about a conformational change. So now information is being transmitted from the extracellular units via the GPCRs towards the intracellular part. Now these GPCRs it form seven units. It's just like it's normally called GPCR or 70Ms because of its unit. It undulates seven times within the intracellular part of the cell. So now let's talk about how information is being transmitted from the ligand then down towards the receptor. So once information comes from outside, you get inside. What happens is the GPCR it binds to the G protein. Now, the G proteins they have two units. That the G protein alpha subunit now. So they have two subunits. We have the stimulatory unit and the inhibitory unit. So it actually binds to the stimulatory unit. So this stimulatory unit, what it does then, it leaves this stimulatory unit, that GAS from this slide here, then since they are heterotrimeric, then this G alpha unit, it then leaves the trimeric, it then leaves this trimeric family, then it binds to adenylate cyclase. Now, this part is the beginning of this second messenger, which is the cyclic AMP. Now, for the second messengers, we have three major second messengers. We have cyclic AMP, we have cyclic GMP, then we have the phospholinositide, and this phospholinositide also has a calcium. Also, calcium is also part of that pathway. So once the G protein alpha subunit once it binds to adenylate cyclase, what happens is adenylate cyclase it changes it changes the ATP. It then changes ATP to cyclic AMP. So that's how the second cyclic AMP. That's how it happens. Then go to second slide, please. Continue with the second slide. Okay, I'm done with this one. Continue with the other slide. Okay. To continue with this slide. Okay, let me just read this slide. So, GPC are principally synthesized in the rough endoplasmic reticulum. They are the receptor that signals via G proteins. They makes up the largest receptor family They are called the 7 transmembrane or serpentine receptors. So, receptor polypeptide chain snakes across the plasma membrane 7 times. Like what I said earlier, they function as monomers, dimers, or oligomers. Go to the other slide. So the first round of signaling initiated by GPCR is a uniform. So active receptor catalyzes the exchange of GDP for GTP. Now the thing is, at the resting state of this heterotrimeric subunit, this alpha G protein alpha subunit, it actually attaches itself to GDP. So once it leaves the other family, that is beta and gamma, then before it leaves, it exchanges GDP for GTP. In fact, that's the reason why it actually leaves those other guys, because it has more energy now. So it exchanges GDP for GTP. So the receptor cytoplasmic terminal tail contain numerous serine and threonine residue whose hydroxy groups can be phosphorylated. Go to the next slide, please. Oh, that's it. Okay. Can, continue. Let me see this slide. What's there? Wait, there's a whole lot of that. So, I will talk about the cyclic GMP. So, the cyclic GMP is almost the pathway. It's almost similar to the cyclic AMP, but the thing is, the cyclic GMP has two. They have two molecules that actually brings about that pathway. Now, for the cyclic AMP, the major enzyme there is adenylate cyclase. While for the cyclic GMP, the major enzyme here is guanylate cyclase. So what happens here is the AMP, that is atrial natriuretic peptide, which is located in the plasma membrane. It actually stimulates guanylate cyclase in the plasma membrane. So while the nitric oxide, which is located in the endoplasmic reticulum, is what actually stimulates the cyclic GMP within the cytosol. So it's being stimulated by two molecules there, which are AMP and nitric oxide. So that then from there, once AMP is being stimulated, once AMP stimulates guanylate cyclase, then guanylate cyclase actually converts GTP to cyclic GMP. So same with oxide, it stimulates guanylate cyclase in the cytosol. Then the guanylate cyclase then converts the GTP in the cytosol to cyclic GMP. Now there is another third pathway I talked about, which is the phospholinositide. So for this phospholinositide family, what happened is there's an enzyme which is phospholipase C. Phospholipase C, it actually split phospholinositide 4,5 bisphosphate. It split phospholinositide 4,5 bisphosphate to two subunits. So it split it to phospholinositol 145 triphosphate, and the slide. Include a lot in this slide. Uh, Talking about this one. So it split it to inositol 145 bisphosphate and diacylglyceride. So it split it to two units, diacylglyceride and phospho 145 triphosphate. So what happens then? IP3 is then converted to calcium. Then, since there's elevation of calcium within the system, then that the elevation of calcium then makes the calcium to bind with camodulin. It binds with camodulin, then other reactions continue. Why For the other part, this diacylglycerol actually is being converted. Diacylglycerol is being converted to I think phospholipase, phosphol, phos, phosphokinase C. Then diacylglycerol is being converted to phosphokinase C. Then from there, other reactions continue. Then the regulatory part for this diacylglycerol is either diacylglycerol can either be phosphorylated to phospholipids, or it can be deacetylated to arachidonic acid. So that's for the regulatory part for this diacylglyceride. Then for the first part, which is inositol 1,5 triphosphate, it can be phosphorylated. It can be phosphorylated. So that is how those two pathways are being controlled. Why for the cyclic GMP? For the cyclic GMP, it can either be hydrolyzed. What happened is for the hydrolysis. Hydrolysis, it can be hydrolyzed, and how does the hydrolysis can happen through the conversion of GTP to GDP. So that's how hydrolysis happen. It can happen through conversion of GTP to GDP. That is reducing the energy of the system. So that's one way it can be controlled. Then also, like as I said G protein alpha sub unit. It has two parts, which is the stimulatory part and the inhibitory part. So the inhibitory part can either inhibit adenylate cyclase. So once it inhibits adenylate cyclase, then there is no conversion of ATP to cyclic AMP. Then also it can also be phosphorylated. Also, it can also be phosphorylated. So these are the three major ways that these second messengers as in cyclic AMP can be regulated. Then for another major receptor, which is after reaching an initial peak level, G protein responds to hormonal stimulus fade away with time, even in the presence of agonists. Yes, there are some instances where even though there are agonists attached to the receptor, the systems continue to fire. So why this happens is just to maintain homeostasis, just to maintain homeostasis. So now this process is called desensitization, and it could be rapidly reversible in some cases if the second messenger is provided quickly before the termination of the first one. So this second messenger response is similar of the initial stimulus. So multiple mechanisms contribute to desensitization of GPCRs. One very well understood is phosphorylation. So go to the next slide, please. Then beta adenoreceptor agonists not only activate G protein, it also recruits and activates a family of course, I said it, okay, I haven't said this, GROKs. Yeah, it activates GROK. GROK is another it's the major regulatory enzyme here. So continue. Okay, for GRK, it phosphorylates serine and threonine residue in the receptor cytoplasmic. Now, this GPCR kinase, it actually it phosphorylates GPCR. That's the threonine and serine residue of GPCR in the cytoplasmic. That's what GRK goes to phosphorylate. So once it does that, it diminishes the ability of activated beta receptor to activate G protein alpha subunit, this GS. That is the stimulatory part. So increasing the family affinity to increases the receptor affinity for binding a third receptor. Now, what happens is when this GRK binds to the GPCR, the cytoplasmic tail, That means it hinders any reaction between this GS protein. So what happens as it hinders it, there's another third protein which is the beta arrestin is being stimulated. So what happens? This beta arrestin it goes and it arrests that part, that tail. So that's what it does. So go to the next slide, please. Then phosphorylation by GROK. So terminated upon removal of agonist leads to dissociation of beta arrestin. Now, once the agonist is being removed, that's how this GROK activity is being terminated. So we know that is. Let me clarify this. But once beta arrestin binds to that cell, that means it stops it just stops any further downstream reaction. That is any reaction that will accelerate or that will produce a second messenger pathway. That's what this beta arrestin. It's just like a plug. It just goes to plug the threonine and the serine area there. So cellular phosphatases removes phosphorylation, so reversing the desensitization state, so allowing activation to occur once again upon another encounter with another agonist. So is there another slide? Okay, I think beta arrestin can accelerate endocytosis of adrenoceptor from the cytoplasmic tail. In some cases, act as a positive signal and transducer if activated through a different mechanism. I think this one is a different thing entirely. Continue. Uh, so continue. It's slightly left. Okay. So intracellular second messenger cyclic AMP mediates several hormonal responses, mobilization of stored energy, conservation of water by kidney, calcium homeostasis, increases stress and contractile force of fat muscle, regulates the production of adrenal and sex steroids, regulates relaxation of smooth muscle, regulate many other endocrine and neural processes. Continue. So it carries out a function via stimulation of cyclic AMP-dependent protein kinases, comprised of a cyclic AMP binding regulator, dimers, and two catalytic chains. So MP binding regulatory. When cyclic AMP binds to the aerodimers, active C chain are released to diffuse through the cytoplasm and nucleus where they transfer phosphate. I think I talked about this. I think I go to the next slide. Intracellular actions of that are terminated. Go to the next slide. Okay, so milrinone, a selective inhibitor of type 3 PDE, is so expressed in cardiac muscle cells, so used as a supportive drug for treatment of acute heart failure. Then competitive inhibitors of cyclic AMP depletion. Uh, so for this one way, one of the instances is the way caffeine, theophylline, and other methyls and xanthine products is the way they act. The way they act in the system. It's a competitive inhibition of cyclic inhibition. But continue, please. So begins with the hormonal stimulation of phosphorinositide hydrolysis, binding to the G proteins, NIMP receptor. Meanwhile, others bind to threonine, okay, tyrosine kinase. So membrane enzyme phospholipase split. I talked about this. Continue. Confined. Okay, let me just That is glycerol confined to the membrane activate calcium sensitive protein kinase called protein kinase. That is PKC. Okay, continue. Okay. IP3, which is water soluble, actually diffuses through the cytoplasm to trigger release of calcium by binding to the ligand-gated calcium channel in the limiting membrane of internal storage vesicle. So, high concentration of calcium causes it to bind to carbon, like what I said earlier. It regulates activation of other enzymes, including calcium-dependent protein kinases. Continue. So it's inactivated by dephosphorylation. So RP3 is inactivated by dephosphorylation. While the acylglycerol is being phosphorylated to make the phosphatidic acid, converting it back to phospholipid. I talked about this. And that acylglycerol, it's being it's either it's converted to arachidonic acid by it's being deacetylated to arachidonic acid. Continue, please. So calcium actively removed from the cell, yeah, through calcium conductor is being removed from the cell. Then lithium ion used in treatment of manic depressive disorders. So it's actually operate via this pathway. Continue, please. Then it's something that continue. Next slide, please. Okay, that increased cyclic GMP concentration causes relaxation of of vascular smooth muscle, which caused by kinase mediated mechanism, result in dysphosphorylation of myosin light chains. So, in the smooth muscle cells, cyclic GMP synthesis can be elevated by two transferrin. I talked about this, ANP and NO. And next slide, please. So, AMP now a blood-borne peptide hormone stimulate the transmembrane receptor bind to the talked about this. Next slide. I talked about this. So, yeah, so useful vasodilating drugs. So nitroglycerin and sodium nitroglyceride so actually operate through this cyclic GMP. So they are used in treatment of cardiac ischemia and acute hypertension. Yeah, so they act by generating or actually this nitric oxide it acts by mimicking it mimics I think acts by the generation of mimicking nitric oxide. Yeah, so these two drugs they act by mimicking nitric oxide. So that's how they control acute hypertension and cardiac ischemia. So then other producers dilation by inhibiting PDEs, interfering with the metabolic breakdown of cyclic GMP. Yeah, continue. So sildenafil is actually used for the treatment of erectile dysfunction and pulmonary hypertension.